Hello, everyone, and welcome to Know4Nano Workshops. I would like to thank you, all of you, to participate in this event organized within Know4Nano project. My name is Vasa Radonić, and I'm Assistant Director for the Sciences in the BioSense Institute, and also the project coordinator on Know4Nano project. On this workshop, that is event that will last for 5 days. So generally, we will organize different lectures related to biosensor, microfluidic, food safety, pathogen detection, but also some lecture related to technology transfer and soft skills. So later on, I will give you more details about the workshop, but first I would like to present the Know4Nano project. Project is entitled Knowledge and Skill Transfer for the Application of Nanotechnology Biosensor for Foodborne Pathogen Detection, or shortly Know4Nano. It's financed by European Union under Horizon Europe program. Most exactly, the topic is twinning bottom-up approach. So the project partner beside the BioSense Institute, where we are now, is the Catalan Institute of Nanoscience and Nanotechnology from Barcelona, Spain, group led by Professor Arben Mercozi, with strong expertise in nanotechnology application, in biosensor development, microfluidics, and nanoelectronics. The second partner is University of Chemistry and Technology Prague, with strong expertise in material science, material characterization, and synthesis. So the group is led by Professor Sofer Zdenek. And the third partner is a National Research Council from Rome, Italy, group led by Professor Sabato Dauria, with strong expertise in microbiology, standard detection method in food safety, and also biosensing. So the general idea of the project is establish a knowledge transfer platform to improve the excellence capacity of all partners by enhancing the strategic networking activity between them. So in general, the Know4Nano will enable not only the biosense research, but also researcher, but also the all actors involved in the project to acquire essential expertise and skill in different fields, such as nanomaterials, biosensor, microfluidics, and food safety, in order to increase their individual portfolio, but also to boost international research capacity of all institutions. So the strategically project is designed to enhance also the research and management capacity and administration skills, especially of the BioSense Institute, in order to establish the technology transfer office and to transfer research idea to the commercial product in the end. And probably the complementary expertise will lead to develop on some cutting-edge biosensing platform for practical application in farm-to-fork chain. This is illustrative picture how the project is organized. So probably the transfer of some specific knowledge from different partner institutions will lead to develop some kind of portable biosensing device with advanced materials and some lab-on-the-chip platform that can be used in food safety application to detect multiple pathogens. So the Know4Nano will provide multiple impact, first in research and innovation. So it will lead to some new scientific discovery, mostly related to nanomaterials and their application in the biosensor development. Technologically, it should fill and modernize infrastructure of the BioSense and fill some gap in infrastructure capacity, mostly related to material synthesis and the biosensor development. Economically, it should develop a new market for touch biosensing tests that will be developed during the project. And also, probably with this project, we would like to link our research with different business and create strategic investment opportunities for the future. Generally, societal impact will be in rising analysis on consumer on different biosensing application in food production to minimize the foodborne diseases and illnesses. And scientific one is reflected in knowledge transfer and education of new generation of students and young researcher in during the project. Oops. So therefore, the Know4Nano Workshop is organized as a 5-day event, collocate with 2 other projects. One is the Microlab AptaSense that is funded by Scientific Fund of the Republic of the Serbia, and also Living Lab Green Studio, also financed through the IPA project and European Union. And probably their team will join us tomorrow on technology transfer section, but also one additional team will join us in the Friday related to soft skill transfer, and it's also participating in another twinning project that's BioSense realized. It's called Sonata. So the event is organized as a 5-day event. So we try to somehow split different sections, but probably to organize 40 different experts in the field to provide lecture on the same day. It wasn't so possible. So therefore, probably you will have some mix on different lectures during these 5 days. But mostly in the event, 40 different experts from academia and industry will participate and provide lecture related to their expertise. We'll also organize one roundtable related to technology transfer tomorrow, and event is organized and combined one. So most of the participants will be presented live, but we also have some lectures all around the world that will join us to provide online lectures. First of all, I would like to thank our partners to help us in the preparation of this event, and probably their willingness to invite different people and different colleagues to provide some specific lecture that will be interesting to all audience. Also, I would like to thank our external expert and guests that will join this event, and also advisory board that will also provide the lectures during this event. And also I would like to thank the people that organized this event. So probably the project co-coordinator Slavica Savic and the dissemination manager Isidora Čolić, as well as the Miljan Mladenović and Aleksandra Pavlović that help a lot with preparation of different materials, web, etc. And also Ivana Milošević and Milica Govedarica with some technical things. So what is the idea of this event? So probably generally we will start from the different lectures related to biosensor development, the application of nanomaterials in the biosensing, synthesis and characterization of materials, and probably with my following presentation, I would like to somehow connect all this lecture and to give some basic introduction to biosensing and their application in the food safety. So therefore, the title of my presentation is based in principles and application of the biosensors. So in simple, the biosensors are some analytical devices that combine biological components with the detector to measure some specific biological materials. So the two mostly used sensor that you can buy on the market is the pregnancy test and the COVID test. So probably that's the simplest type of the sensor based on lateral flow, when you can recognize the positive or negative results. But a little bit complicated one is the sensor for the glucose. So generally, it's use some kind of analyte. In this case, it's blood that need to be put on some testing strip with some biological component that react with the glucose in the blood, and then at the end you measure that with some kind of detector. This type of sensor, in comparison with pregnancy test or COVID test, can give you the quantitative results of the analysis. So therefore, we can tell that the biosensor is composed on several parts. First one is the bioreceptor, which role is to recognize some specific biomolecules in the analyte that you would like to detect. Then we have the transducer, then convert this biological signal to some recognizable signal. And at the end, we have some kind of readout devices. So as a bioreceptor, different kind of biomolecules can be used, such as nucleic acid, enzyme, antibodies, aptamers, peptide, flagellase, or many other. So, and their role is to catch some kind of biomarker in the analyte that can be water, saliva, blood, or other type of biological material. Another important component is biosensor transducer. So, and their role is to transform this biological signal to some readable signal using different detection principles. For example, it can be electrochemical, optical, magnetic, piezoelectric, thermal, and can work on some other principles. And at the end, we have some kind of electronic and readout system that transform these results to some signal that can be recognized by end user. Generally, it composes some amplifier, some noise eliminator cycle, processor, display, etc. So besides transducer and bioreceptor, we can also use some biocompatible layer or nanomaterials to improve the sensor performances and transduce principles. Let's see how the biosensor operates. So generally, we'll start with this, the simplest one, also called lateral flow or colorimetric sensor. So generally use some kind of analyte that you put on some specific part of the sensor that's called pad. And through the capillary force, this fluid flow through the complete sensor. So, based on what would you like to detect, at the end we have the two control line where the probably antibodies or some biological elements are already functionalized. So, and based on analyte that you would like to detect, if there any complementarity between these antibodies and the biotarget that you would like to detect, they will attach to that surface, and probably they are sorry, they are functionalized with some colorimetric dye that will react with the changing of the color. So probably in this type of sensor, generally two type of antibodies are used. One, primary antibodies for the analyte that is specific, some specific analyte that you would like to detect that serve as a control line, and the antibodies that use for the testing that detect presence on some kind of biomolecules inside the bio, is on the test. So generally, I'd like to move to another slide. I will do an arrow. So another type of sensor work at another principle. So generally, some electrochemical use some electrode on which we can functionalize this different type of material, such nanocomposite or different nanoparticles, 2D or 3D nanomaterials, which is later on functionalize with different biorecognition elements that catch the target from the analyte. And what is the role of these nanomaterials? Why we need it? So generally, nanomaterials in the biosensors are used to enhance biosensor performances in terms of sensitivity, selectivity, going on, and stability. So, and mostly common nanomaterials used in biosensor include carbon-based nanomaterials such as graphene, carbon nanotubes, 2D materials that are mostly used to improve electron transfer and provide structural support for biomolecules. Besides that, we also use metallic nanoparticles such as gold, silver, or platinum that can be additionally functionalized with the biorecognition elements and mostly used in optical biosensor to enhance some surface plasma resonance, etc. Also, beside that, we use the quantum dots, metal organic frameworks such as the porous materials to enhance analytical binding and catalytic reaction, and also nanostructure polymers such as polyaniline or dendrimer to improve immobilization on the sensor surface and increase the sensor sensitivity. And what is the application of the biosensors? Today, the biosensor find application in different fields, such as environmental monitoring, food quality monitoring, and food safety. So toxin detection in healthcare applications, such as drug discovery, detection of tumor biomarkers, and many others. And mostly they are used to detection of different kind of contaminants such as heavy metal, pesticide, fertilizers, phenol compounds, pathogens, or in antibiotic research. The project is mostly oriented to the food safety. So and the food safety control is essential to prevent different foodborne illnesses that's caused by presence of different pathogens in the food. Also to ensure the public health, maintain consumer trust, and also to comply with some regulational standard. And the biosensor can help in that area to detect different contaminants, as I mentioned, pathogen, toxin, and reduce the risk of some kind of outbreaks. Let's see, probably what is the application of the biosensor and what we can detect with the biosensor in the food safety. First of all, as I mentioned, that's some kind of pathogens such as bacteria like Escherichia coli, Salmonella, Listeria that can be found in the meat product, vegetables, but also in some milk product. Also, it can be used to detect different toxins, such as mycotoxin or alpha toxin or some marine toxin. It can be used to detect the pesticide residue analysis, so to measure some harmful pesticide level in fruit, vegetables, and grain. Also to some allergen detection, nutrient analysis, and many other applications. Why we need biosensor? So generally the standard detection method that are used to detect all these contaminants require some specific laboratory condition. They are very slow, require expensive instrumentation, trained personnel, and they have many other disadvantages. On the other side, the biosensor can provide rapid, low cost, and accurate solution that are greatly needed for in-field analysis. So, on the other side, the market also needs some low-cost and fast diagnostic tools for in vitro diagnostic to be used by unprofessional and non-trained person to obtain the results directly on the site. And probably the potential user will be some inspection services, food industry producer, and even the consumer to test the food that they eat. But some analytical analytes or biomolecule is not easy to detect in some specific samples. They require some specific extraction protocol and complex procedure in order to be detected by biosensor. Therefore, modern biosensing device integrate complete laboratory in a single chip, called also laboratory on the chip. So with the development of technology, probably with the advance in micro and nanotechnology over last 2 decades, the microfluidic become a key component to develop some kind of portable sensing devices in the platform. And generally, the microfluidic is the area of research that deals with the manipulation of the fluid and the microscale in the network on micro and nano channels. And today, it finds application in different diagnostic, drug discovery, biological research, etc. Advanced microfluidic today integrate a complex function in the single chip, such as DNA extraction, amplification, mixing of the fluid, particle separation. They also integrate different sensors and actuator, reagent storage, and many other components. And on the 2 examples, I would like to show you how these complex systems look like. So probably this is some kind of autonomous system that take the sample from some kind of cultivation, they take cell cultivation media, they measure its pH, mix with some buffer, then mix with some enzyme and lead this fluid to the sensor. Also, some more complex solution integrate different kind of components in the same chip. They integrate some kind of incubation chamber. They also perform some cell lysis, hydrolyzation, extraction, hybridization of the biomolecules on the sensor surface, and also some kind of optical detection, everything on the same chip. In that case, this complex system offers some kind of device that can perform all analysis on the same chip. That's also in the same time simplify detection, save reagents, eliminated the need for specialized and trained personnel to help to operate with this device. So with this some kind small introduction, I would like to point you with different aspect of the biosensor development we'll address during this workshop. So probably we'll talk about the biosensor, the microfluidic, different detection techniques, also about the new materials, their synthesis, characterization, and application. So we'll have possibility to learn different standard detection method, but also how to test and validate our biosensor, and we'll have possibility to go deeply into food safety aspect as well. And also the part of the workshop will be related to technology transfer and improve the skill of the researcher in order to help you to see the all possible option, how to turn your research idea into some commercial product at the end. So how to improve your knowledge and skills to be ready to turn your idea into the patent or successful proposal, and how to use your funds to further develop that on the idea and launch the product in the market. Probably you will have possibility to learn from experienced researcher and expert all around the world. So probably I wish you successful event, and thank you all of you to join to this workshop. If you have any question, I'm open.